We've been talking about evolutionary biology a lot, and we're going to continue to do so, but let's take a brief break and talk about some important concepts in molecular biology. Molecular biology is a field in and of itself, but some of the discoveries from this field greatly inform how we view evolution. Today, we're going to talk about the discovery of DNA, or its structure and its function, and then a little bit about different types of cells. As a reminder, this is a quick overview of a couple important concepts in molecular biology, and we will not be going as in depth if you were to take a molecular biology course. I encourage you to find a molecular biology course to learn this more in depth. So let's start with our cell. Here's a basic animal cell. It's, kind of this weird blob shape, but we have a bunch of different cool things inside. The thing we care the most about is that purple ball in the center, the nucleus. Within the nucleus, there are a couple cool things. We discovered chromosomes first. Chromosomes are named colored body, is what that word means, because under a microscope, we saw these super compacted things that we could stain different colors. It took us a little bit to figure out that chromosomes are where the genetic material lies, or where we are storing hereditary information. It was a big debate. At first, some people thought it was proteins and not DNA that actually held all the important information. So DNA was discovered in the 1950s. You can see here's the publication in 1953 in Nature by Francis Crick and James Watson, but it was more than just these two gentlemen who were important in the discovery. Maurice Wilkins and especially Rosalind Franklin did a lot that was very important. This is an X-ray chromatography. So this is a type of X-ray that helps us determine the molecular structure of very tiny molecules, because DNA is, of course, too small for us to be able to observe with our two eyes. So we have to use some roundabout ways. And it was Rosalind Franklin who took this picture, and it was her interpretation that was critical in helping Crick and Watson determine that it was a double helix. But let's talk about DNA, or deoxyribonucleic acid. So DNA is a double helix. So that means we have two strands that are wrapped around each other. Here we can see it is both uncurled and curled, so you can see the structure in two different formats. What's interesting is these strands run in the opposite direction. On the molecules itself, we have a 3' end and a 5'. So if you look at our ladders here, one strand has 5' on one end, but the other strand has the 3' end opposite of it. You'll also notice in this twist here, it is an uneven twist. We have a major groove, or a big gap, and then a minor groove, a smaller gap. So whenever you see those representations of DNA out there, but they're an even twist, that's not right. Another important thing is the twist is to the right. So, which one is a right twist? So here, picture A is our right twist. To figure it out, take your right hand, put it in front of you, and twist to the right as you move forward, and that is the direction that all DNA should be twisting. So if you ever want to figure out, that's the trick you can use. Deoxyribonucleic acid, we have this sugar phosphate backbone. In our picture here, that dark red line, that is where the sugar and phosphate is. And we have in the center, our nucleotide bases are held together through hydrogen bonds. Hydrogen bonds themselves are not particularly strong, but if you have a whole bunch of them lined up together, and then you twist those strands on top of that, this leads into a very stable molecule. So here we have a little bit more information on the chemical structure of DNA. We have that sugar phosphate backbone. We have these 4 different nucleotide bases held together in the middle by our hydrogen bonds. But why is it important in the first place that DNA is stable at all? Do you know? So DNA structure, a key principle of physiology is that structure is the key to function. The things it can do determine the structure and vice versa. Because DNA is a stable molecule, that made it a good storage medium. It is a good storage medium to contain hereditary material, and that's probably one of the reasons why evolution eventually decided on DNA as the storage medium. Because we need to store genetic material throughout our lifetime, if it's stored incorrectly and has mutations, that's cancer. And we also want to be able to reliably pass on genetic material to our offspring. If we cannot reliably pass on advantageous traits to our offspring, then we're never going to end up with evolution the way we know it today. But let's talk a little bit about those four different bases, or our nucleotides. We have two versions. There are purines and pyrimidines. Purines have two rings, and these are adenine and guanine. Pyrimidines just have one ring, thymine and cytosine. You can see their molecular structure here. Also, we have uracil because RNA has a slightly different structure. And on top are purines, again with those two rings, and pyrimidines on the bottom with just a single ring. What's really cool about DNA is the complementary base pairing. A purine always pairs with a pyrimidine. We have adenine goes with thymine, and guanine goes with cytosine. And that is where our hydrogen bonds are. And we can zoom in a little bit here. And you'll notice the reason why it's this very, very consistent base pairing is guanine fits with cytosine really nicely, and adenine fits with thymine. This is really important because this consistent base pairing means we can make a reliable copy. But the other important molecule we have is RNA, or ribonucleic acid. This is a little bit different than DNA. This is frequently found in a single-stranded form. We also have uracil instead of thymine, and now we have lots of different types. So many different types of RNA. I don't follow RNA research that much, but I feel like every time I check in with the field, there's a new type of RNA that we're talking about. That's just how much we're discovering here. And the other really cool thing about RNA is it can actually self-organize into a three-dimensional structure. And it does stuff. RNA has actual function. DNA is really boring. It sits there and it stores information. RNA does stuff with that information. So that is why, and here are just a few examples of the vast arrays of different types of RNA: RNA, tRNA, microRNA, rRNA, sRNA, cyst, incRNA. Some of these, I don't even know what they are. And all of these things have a different function in our cells. Because RNA has the ability to carry out different biological functions, we think that RNA actually preceded DNA in the evolution of the world. So here shows that RNA, because it has the ability to self-replicate, not very well, but it can, that is probably the very first form of life that evolved. Eventually, DNA became the template because it is more stable and a little bit more reliable as a storage medium. But let's talk about how we use it. So, in our chromosomes, DNA is a very small strand which we wrap around nucleosomes, or wrap around histones to form nucleosomes, and then that coils into chromatin loops, and those chromatin loops curl up into a chromosome. So our chromosomes here are super compacted DNA. Now that we're a little bit familiar with the structure of DNA, let's talk about the functions within our cells. The central dogma of molecular biology is the most important thing you can get from molecular biology, so let's make sure we understand it. That just means DNA is transcribed into mRNA, that mRNA leaves the nucleus and goes out into the cytoplasm of the cell, and through translation is turned into a protein. Here is a slightly different visualization, and now you can see that translation happens in the ribosome of our cells. Gene expression, that is just the translation process where we are making proteins from that mRNA transcript from our gene. And we can look at it a little bit more closely here. Our transcription, of course, happens in the nucleus. We are making a transcript of mRNA from our DNA that leaves our cell, and it goes into a ribosome where we do our process of translation. Sometimes there's a little bit of modification that happens in between some of these steps, but that is the basic idea of how we go from DNA to a functional protein. And how we do this is through the genetic code. This is mostly universal. We have a group of 3 base pairs which form a code that relate to one of the 20 different amino acids. And we call this our codon. That is the sequence of 3 nucleotides of DNA or RNA, depending on what you're talking about. This is a unit or a word in our genetic code. And here you can see in our ribosome, we have our tRNA brings an amino acid. It matches up with a specific section of our mRNA transcript and builds that amino acid chain like beads on a string. But DNA can do one other thing. It can replicate itself. So if you've ever heard this pickup line, I wish I was DNA helicase so I could unzip your genes, that's what it's referring to. DNA helicase is the enzyme that unwinds DNA so we can go in and replicate both strands, and now we have 2 identical copies. This is the reason why that complementary base pairing is so important, because to do this, we need to be able to reliably create a new copy from just half of the strand. We can do it through this. In mitosis, we'll just get an exact copy, or we can also have something called meiosis, where we end up with 4 daughter cells that only have half of the genetic material in our gametes because they're going to combine with the gametes of another individual. And we want to have half of our material because if we both add both together, now we have an offspring that has twice the amount of genetics. In meiosis, a couple of cool things can happen. Crossing over is the exchange of genetic material between homologous chromosomes during the first prophase of meiosis. It's one of the ways we can get genetic recombination. Here is a diagram. So we have our homologous chromosomes line up. They can cross over and literally just swap some pieces. We generally like to visualize this through different colors because now you can see some of the chromosomes are a little bit of a patchwork. And they have some of one chromosome and some of another. And this is how we can swap different genes and get new combinations amongst alleles of different genes. Meiosis is one of the, or there are a couple different ways we can increase genetic variation within meiosis. First is that crossing over, which we just talked about. Second is random assortment. We don't know which allele you're going to get. And of course, if mutation happens during meiosis, we can have something brand new in our offspring. It's also important to talk about the different types of cells we have in our body. We have 2, somatic cells and gametic cells. So here, as we undergo development, our somatic cells, they create all of us, everything you can see. But those are not going to result into offspring. The ones that will result into offspring are our gametic cells. However, our somatic cells, even though they won't be making any children for us, they do have a really interesting development cycle. As they develop, they actually lose the potential to turn into different types of cell, and they differentiate into a very specific type, and they get more and more differentiated as development happens. So in this example, eventually we have a circulatory system cell that it can only turn into a part of the circulatory system. And eventually we'll have heart cells that can only turn into other heart cells and not blood cells. Same for the nervous system and the immune system and other body systems as well. And we have a couple different names for this. Totipotent is when you can turn into everything. Pluripotent is most things. And then we're going to be more and more differentiated. So that helps us understand one of my very favorite memes, Cell Career Day. If you work hard and undergo induced pluripotency, you can be anything you want to be. And this is the principle behind stem cell research. We are taking fully differentiated adult stem cells and we're trying to somehow turn them back and make them pluripotent again so they can become many different types of cells. So, now that we've talked about some important concepts in molecular biology, can you explain what's the structure of DNA and how is that important to its function?